Let's see. Here we go. Is that full screen and you can hear me all right?
We can.
Okay, great. So I'm thrilled to be back at Stennis and at the Marine Sciences University of Southern Mississippi. I had so many great visits. It was my first faculty offer, and I wasn't right for my family at the time, but you know, there's so many amazing people that have come out of your area that are still great friends of mine, and so I'm so impressed with the work being done there. But I ended up here in New England, and you can see from this MODIS image, I'm going to talk a little bit about how we've come towards hyperspectral remote sensing, and Long Island Sound in particular, which is this little sliver of water, one of the most domesticated water bodies in the United States. And our little sliver of the world is the University of Connecticut here. My background, just so you know, I did do biological sciences, and then I went to California, and I like these little graphs because I study ocean color, and you can see the dramatic changes in the color of the waters. I went to UC Santa Barbara and got my PhD in geography. You can see this green lagoon, and you can see the surf and the greener waters around Santa Barbara there, compared to that deep Monterey water. And then I was a visiting scientist at University of Miami, which has those optically shallow kind of aqua waters, and ended up at Moss Landing Marine Labs as a postdoc, where you can see those deep Monterey waters as well, and some coastal lagoons, and Mbari, which is down further in this picture. And now I'm at beautiful University of Connecticut. Jiao Dong was just here, and I welcome any one of you who wants to come visit me. My office is this building here, right on the shores of Long Island Sound. So this is my lab website. You can take a look. I need to update it a little bit, but as always, our work has involved sort of a diversity of remote sensing, a lot of work on benthic remote sensing like seagrasses and corals, which I'm not going to talk about today, but you could invite me back and I'll do that another day. I've kind of focused on the backscattering of bright surfaces, and I have, if you look at my CV, it seems maybe a little bit disparate. I certainly have a lot of papers covering diverse topics, from whitecaps and foam to sea ice to floating vegetation. I've been doing a lot of work on microplastics lately, sediments, cyan and red tides, seafloor. And so today, though, I want to sort of summarize more of the phytoplankton work that we've been doing and relevance to some of the upcoming missions. Most of you know the oceans are the lungs of the planet. You know, people think the Amazon is really the lungs of the planet, but we know the oceans, you know, have greater than 50% of the productivity. But, you know, what always strikes me is it says equals or exceeds, you know, like that's kind of a big difference. Does it equal or does it exceed? So there's still so much uncertainty in our net primary productivity estimates for the oceans in particular. And I'll give you a few reasons why we have those differences. Phytoplankton is very small in the ocean. It's very diverse. And we have this kind of inverted pyramid where we have a lot more consumers than producers in the biomass. And we have this phytoplankton diversity of different types. And certainly these are contributing different amounts, I'll show you, of productivity. So the ocean food webs are complex. Phytoplankton, we call them at the bottom of the food web, but they're not all the same in terms of food quality and carbon uptake. And right now, our remote sensing models kind of treat them all as a box of phytoplankton that's doing some of the same things. And our modelers are kind of trying to be more complex and give functional types. And we know that from new technology like the imaging flow cytometry, this is from Alison Chase, some of her work looking at phytoplankton sizes and structures, that they're much more diverse and they're doing different roles in the ocean. And so we need to sort of step back in our remote sensing and try to get more nuanced in what we're looking at from the world's oceans. I mean, not a lot of people think about food quality, but this is a nice paper from 2017 that took a transect across the Southern Ocean and up towards Australia and looked at the phytoplankton taxa. And as you can see, the different groups for each station contribute different percentage of the pool of phytoplankton, from chlorophytes, cryptophytes, diatoms, dinoflagellates, getting to more of the smaller as we get closer into that middle of the gyre, in that hotter water there. We get more of the smaller prochlorophytes and Synechococcus, but you can see any one station has a pretty big diversity. And there's vast differences in food quality in the food web, so how these things go up the food chain. And then in terms of flux of carbon in, we like to treat them all as this one box of phytoplankton that takes up carbon. I was part of this really big airborne Southern Ocean study called ORCUS, and we flew aircraft all across the Southern Ocean. And you can see on the right that the models are all in perfect agreement as to whether the Southern Ocean is a source or a sink of carbon. You can laugh. No, they're not. Obviously, each model has a very different, I mean, this is where we get down to the crux of the matter, is we just don't understand how these processes are contributing very well, especially in the Southern Ocean that has an inordinate role in the carbon flux on the planet. And so we, again, are trying to investigate some of these processes. I have a new grant that's interdisciplinary, looking at phytoplankton and sea ice and dynamics down in the Southern Ocean. My postdoc just deployed and is sitting in a hotel for 2 weeks, waiting to get on a ship down in Puerto Natales. But from that study, you can really see the ocean breathe. And I don't know if you have ever seen the ocean breathe, but I love this footage. On the right, the red is oxygen coming out of the ocean, and the blue is carbon dioxide going into the ocean. And you can see as this plane gets close to the sea surface, you can see the oxygen coming out and the carbon going in there. There's this inverse dynamic in real time. I'll see if I can play this without hitting that silly button. All right. So just because I think it's very cool to see the ocean breathe, because I'd studied the ocean a lot and actually never seen it fully breathe before. So what we've been finding in the Antarctic, if this is the uptake delta P partial pressure of carbon dioxide as a relationship with chlorophyll, you can see there is kind of a relationship. That's how we model it today. We can say there's the less amount of chlorophyll, the more comes out of the ocean, the more chlorophyll, more goes into the ocean. But you can see so if you pick chlorophyll 10, which is a lot of chlorophyll, sometimes it's not doing a lot of carbon uptake, and sometimes it's doing a lot of carbon uptake. And that variability, we don't really fully understand, but some of it is driven by phytoplankton diversity. And so this is a paper in the Southern Ocean from Brown et al. In 2019 showing that diatoms at the same level, the circle is the chlorophyll amount, are taking up much more carbon than cryptophytes when they bloom in the waters at those same circle sizes, do much less photosynthesis and carbon uptake. So the goal is to sort of understand a bit more nuanced on what is the biodiversity of the world ocean. And right now we sort of do these very crude sort of correlative maps of phytoplankton from space, where we have these one dominant picture, and it's very much based on assumptions about the phytoplankton correlating with chlorophyll. And as I showed earlier, but each one of these spots, even one that's dominated by diatoms or dominated by nano, is a very complex assemblage, a fraction. There's many things going on in this water, and certainly this one dominant group can't really explain that. And so we're trying to move to more fractional compositions of different groups so that these things we can understand better across the oceanscape, and then put this into biogeochemical models that incorporate phytoplankton groups, and we can start to understand the carbon dynamics as well as the functional types in the ocean. And so how do we do this from space? Well, we know the oceans are many, this is from Ryan Vandermeulen's 150 Shades of Green. But we know that the oceans have many colors and types that we can see with our eye. But we know also that there's a lack of spectral information at key wavelengths, and it's limited many of the approaches for trying to get at biogeochemistry. We just don't have enough information. And so we put forth the idea of getting to hyperspectral data. And you can say, I wrote a paper recently, I'll show you how the hype of hyperspectral. Well, many people say we haven't really made the case for hyperspectral. So how many bands do we need? Some people say we just need several multispectral bands, just a few more bands. So these are all the bands you may see. And some of the sensors like Landsat are these big broadband RGB sensors. And then we have our MODIS and VIIRS that have sort of these big gaps in the green leading to the far red. And then we're getting more filled in with bands like ULCI that have select bands. In other sensitivity studies, like CEOS did simulated spectra, and they concluded that a sensor with 26 bands between 380 and 780 was probably adequate for retrieving aquatic ecosystem variables. And you can see they used the simulated data set. The bottom here is wavelength. For those who don't look at that, there's blue from 400 to 500, green 5 to 6, red 6 to 700. And this is remote sensing reflectance. So, this is a really green peaked water in red and all these black variables. Now you can say, well, we can simulate all these data sets, but I'm going to show you some real data, which is why we sort of need to put satellites up at the end of my talk, just spoiler, from HYCO that looks nothing like any of these simulated spectra. So even with this, with simulated spectral bands, we still can't always come up with real world remote sensing reflectance. There's still many things that we don't understand. And so if you look at requirements from what we do think of as average spectra that might be simulated, you can start filling in bands based on different needs and importances. And so at some point you start to say, well, which bands should we, and maybe we should go to hyperspectral data. So I'm just going to go from this point here where these studies trying to say how many bands to, we are putting up satellites that have sort of the whole rainbow, like most of you have heard of, hopefully PACE, the plankton, aerosol, cloud, and ocean ecosystem, which is going to try to fill in this gap that we have in all these other bands and hopefully see things that we've never seen before and help to tease out these phytoplankton pigments in absorption and get at things like carbon and fractional composition. So this is a review that I recently produced with many great colleagues looking at the sort of science and resources and outlook of hyperspectral remote sensing. And this is a figure from that. You know, in the past, we had all these gray sensors that had several bands and different revisit frequencies on the y-axis. And the size of each dot is related to the pixel size. So we've had these 1 kilometer to 500 meter sensors with a few spectral bands. And now we're sort of moving to this more hyperspectral world. And this is going to really revolutionize how we look at the ocean in the next decades to come. We've had sort of a suite of sensors, Hyperion, CRISPR-Proba, and HICO, and I'm going to show you an example with HICO, which was on the space station, which was about 100 meter, 100 bands and about 100 meter pixels. And now we're on the cusp of, we have Prisma up and DSIS, and then SBG coming online, and CHIME, and then of course PACE, which is going to be the first global hyperspectral mission to look at the oceans in all of the spectral bands, with many overarching science objectives. But the one I find very compelling is to try to get at phytoplankton functional types and not go beyond sort of this chlorophyll, and also new things that we didn't know before. So I'm just going to cut through this part here. And just so basically, because I'm the science team lead, I have to sort of amp up this mission, which I feel is going to be amazing because we have all these coupled ocean atmosphere inversion models now. So the first time we're going to be getting at uncertainties in the ocean and the atmosphere together. And try to decouple both of these fluids that have signals that kind of interfere with each other. And what's going to help with that is the multi-angle polarimeters. And I know you know Xiaodong's working very hard on some algorithms for PACE. So what I want to talk about a little bit is sort of my journey into hyperspectral from the phytoplankton side of it, is because, you know, you end up studying the water body you live in. I mean, I've been up to the Arctic and the Antarctic and all over the world's oceans, and I end up kind of coming back to my local water body because it's a really interesting piece of water, and it juts out as the most domesticated body of salt water in the Western Hemisphere, according to F. Scott Fitzgerald, so it must be true. The Great Wet Barnyard of Long Island Sound. Here is Long Island Sound. And you can see it has a very urban estuary. There's 9 million people that live along this little estuary, 4 million from New York and all around. We get annual hypoxic events. There's climate change, devastating hurricanes, and very high biodiversity. It also has a huge watershed, and so it goes almost all the way up to Canada. All these little rivers drain eventually down into Long Island Sound. And so, you know, it's very hard to manage, and we can get sort of algal blooms from nutrient addition. But the good news, and so there's very few good news stories. This is one, is that we think Long Island Sound is improving. So if you look in 2001 to 2010, there was the percent of hypoxic years was much greater in a larger area compared to 2011 to 2016. They've invested in new sewage treatment and other issues to shrink the eutrophication of this water body. It still does go hypoxic in the western part. But when they did the report card recently for 2017, I, of course, live in the A+ region over here. And but you can see it, the western went from a F, D+ to B-, and still some problems down here in the East River coming in. But overall, the water quality is improving, but we don't have very good tools to assess this from space, and I'll explain that a little more. Long Island Sound's also warming, which it's hard to know whether the treatment plants or there's changes just due to the changes in the water quality, including warming temperatures. And then, of course, we have a lot of hurricanes that may be intensifying, creating these sediment plumes that are coming out. So this is some work that I've done over the last, I guess, few decades, starting with my grad student, Dirk Oren, who's now at Goddard. In 2012, we worked on some optics through some work leading more recently. Now, Dirk, when we went out and we looked all over Long Island Sound, what he found is that there was some nice ways to divide the water bodies depending on the location. And basically there was sort of the more river dominated parts and then a very green peaked kind of spectral shape. So some with more sediment and CDOM, and then the river plumes that are a bit different in both the New York Bight and Central Connecticut River there. What he also found out is that it was much more optically complex, as you would imagine, compared to NOMAD, with more absorption by phytoplankton, CDOM, non-algal particles, and certainly more backscattering, BBP being, if you look at Long Island Sound, you can't even compare the scale compared to the NOMAD, which is this little black here, much more CDOM, phytoplankton. It's just got a whole lot going on, and we would call this definitely optically complex. So he evaluated semi-analytical algorithms a whole bunch in a 2012 paper, which this is the formula that's currently used for most standard semi-analytical formulations, which is backscattering over absorption plus backscattering. So getting at these inherent optical properties. And so there was a variety of models he tried, listed here from QAAs to the Garver-Siegel Martorena model. And he fit them initially just as they were written. And what you can see here is, strangely enough, QAA 640 with no tuning. That means it's using this band at 640 nanometers. Actually did really quite well without any tuning to get absorption by phytoplankton and kelp stuff and backscattering. So greater than 75%, even 80-some%. And you think, well, what's happening at 640 nanometers that you would do so much better, and why don't we have that band on most satellites? We have it. Sometimes you can get a land band and use that. And that's because actually nothing's happening there. So when we design satellites, we often overcompensate, and we want to make the where these absorption features are, and we also often miss parts of the spectrum where there's minima. And so what happens is, if you have 640, there's not a lot of absorption there, and you're able to parameterize absorption by Gelbstoff and depigmented particles much better when you have this wavelength. So it was like my first eye opener, like, wow. So when we try to make bands, we have to be conscientious of picking parts that don't seem interesting in the spectrum. And so here we have this early work over a decade ago, saying, well, it would be really nice to split up these bands from some motospheres and have this nice 640 band, just because nothing's happening there. And we're able to actually tune a model with very little. I mean, he went on to tune models using the bands we have, but it's quite challenging to do every water body. You don't want to be retuning a model. And that it works so well in these optically complex waters was truly an eye-opener for us. So here we go. We add the 640 band. That's an important band. But what we found is it was much more challenging to get to chlorophyll. The chlorophyll algorithms tend to fail. If you look on the right here, if you took satellite chlorophyll from an ocean color algorithm and you compare it to in situ, and this is a linear scale, keep in mind, it really does poor job of retrieving. Sorry, did I just lose my spectra here. Stop sharing, sorry. Okay, there I am. So what we found is, in fact, we have this nice time series of Motus and Maris, and we also have the Connecticut Deep going out measuring every month chlorophyll. And what you see is this time series from Motus and Maris of chlorophyll, and you can see seasonally, it says we have these giant blooms in, May, June, July, August, and nothing in winter. And then the chlorophyll measured says something very different. It says we have this kind of early spring bloom, and then we have this late summer bloom that's a little lower in magnitude. So clearly, I mean, this isn't a shock. This is optically complex waters. We know those algorithms don't work. So we started trying to figure out how to look at this in a better way. So we often look with fluorescence. So if you look at a fluorescence line height, say from 678 to 667 from Motus, what we found is initially it looked like a big blob, but when we divided it out between winter and summer, the blue being the winter bloom and the red being the summer, that in fact there was a difference. The fluorescence algorithm line height from Motus worked not at all in the winter months. So we developed an algorithm in this paper based on the fact that we couldn't see these winter blooms from fluorescence, and we certainly couldn't get it from a standard OC algorithm. And so we realized that they still produced a red edge at 709, and we came up with this sort of band switching algorithm that worked pretty well. Now, the point of this is that it requires a red edge, which is a 709 band, and it also required fluorescence bands, either 680 and 665. Now, we know Veers doesn't have fluorescence at all, and it doesn't have a red edge, and Motus doesn't have a 709 band either. So only Maris and going into Olchi would. But with this new algorithm, you can see on the right our chlorophyll seasonal monthly from our RBD red band difference algorithm was working quite well on a seasonal cycle to obtain this chlorophyll signal that we saw measured in situ, and we could finally get sort of nice maps that corresponded mentally with what we knew, that there's this big nearshore bloom in February, that it goes into the spring, and this is mostly CDOM dominated, and then we have a late bloom in July, August, more in the western part of the sound, and then it goes back to very low chlorophyll again in the winter months. So we were really pleased with this. I mean, we could start to look at trends. The problem is you can see that the trend ends in 2012. So with Maris, so now we have to begin again with Olchi and hopefully Pace, because we just don't have the bands that we needed to continue this algorithm and really understand the dynamics going throughout the year. That's the top plot, shows that there's a slight declining trend over this time. And if you look at the bottom plot of Long Island Sound, it shows where the trends are. So there's still some increased chlorophyll around, this is New Haven and Stamford, around the cities. But overall, there's a bit less chlorophyll in the main central parts of the sound, and certainly in this eastern part, which exchanges with the Atlantic Ocean. So this is work that we want to continue with Olchi, and it's always simple. If you want to get imagery, you can go to Copernicus and start to look at ocean color from Olchi. And this is the greatest part of it, is Long Island Sound is so important that it actually intersects the edge of every one of their tiles. So this is another wonderful thing about Long Island Sound. It's right there at that interface between their tiles. Anyways, this is some future work, is to start looking at how these trends go. So Now, just to fill in back to this graph I showed you, now this work showed that really we do need a 680 band and we need a 709 band. And so the VIIRS instrument, you know, MODIS is due to be done in a couple of years, you know, and VIIRS doesn't have fluorescence. These are the NASA bands, NASA missions, right? And 709's never been on a NASA mission. So, you know, these are subtleties, and I can say the PACE mission is really going to amplify this red edge signal. We're going to have better gains than MODIS, and probably than any sensor up there right now. It's going to reset the gains so we can really look at this part of the spectrum, which is really important for getting at phytoplankton dynamics in complex water bodies. And so finally, I wanted, like, the third little bit that to this puzzle was we have this beautiful Long Island Sound integrated coastal observing system, which is distributed sort of throughout with these buoys that is tracking oxygen and temperature waves and things like that. And we had some technicians go out to service this buoy back in the far west, that part which goes anoxic, and they found this colored water, you know, the parting of the Red Seas. They were like, wow. They took a picture and came back in some water, and they're like, this was crazy. The sound was bright red. And so we looked at this sample. This is a mixotrophic ciliate Mesodinium rubrum. And so this is not phytoplankton, but this is a ciliate, a zooplankton. And we did the DNA on it just to be certain. Mesodinium rubrum it was. And when it says it has enslaved cryptophytes, which most of you don't know mixotrophic ciliates very well, but what they do is they take their cryptophytes and they put them on their back essentially without digesting them, and they use them for photosynthesis. So they can eat phytoplankton as well as use them for photosynthesis. I wish I had that skill. Oh, here's just some fun facts on this really cool organism, because my true heart is ecology. I do optics, but I love thinking about organisms and their habitats. So just a few fun facts to share, that they actually retain these ingested chloroplasts. And the plastids that are within these chloroplasts can reproduce and remain transcriptionally active. So actually, they're not just waiting till they decay, they're using these and they're able to keep the plastids going. They're also super fast swimmers. They have the ciliate swimming speed record up to about 1 centimeter per second, which is very fast, and they can change their position vertically to access nutrients. When they are in that red color, they've all swam to aggregations of over 1 million cells per liter. And they can contribute significantly to primary productivity, obviously. But because they're a mixotroph, sadly, they're undersampled and underappreciated. They're not included in any physical model. We don't have any idea how much productivity happens by these mixotrophs and how they contribute to the carbon cycle across the global ocean. But we know they occur, and we know that they're doing incredible amounts of photosynthesis. You can just see all those packed chloroplasts on there. So when we had this event, just to further point out, if you looked at fluorescence line height, we had this bright red blob in the western part of the sound. If you were just a novice and decided to look at the chlorophyll product from Long Island Sound, obviously it did not work at all. I already explained that. But you can see here there was something going on with this big bloom down in the western part. And we actually, Kurt Davis was channeling with us, because I didn't request this. We didn't even know, but he did download a HICO image of Long Island Sound at that very time, which was on the space station. It was called Hyperspectral Imager for the Coastal Ocean. It was sort of our first prototype sensor put up in conjunction with, you know, your folks down at Marine Sciences, Bob Arnone and et al. And it had greater than 100 channels and 110 meter pixel resolution. So we were like, wow, this is a strangely fortuitous, wonderful coincidence. And so what you can see, this is a spectrum in black from that HICO sensor, which we had atmospherically corrected, compared to MODIS in the background, which is, and its few bands, which is the dotted line. And what you can see is it doesn't look like any of those simulated spectra I showed you on those first few slides that we put in all these models of spectral bands. And it looks very interesting. You're like, what is this? These peaked bits to this spectrum. So if you look at the MODIS in more detail, that's this top panel, and over the average from this region, a fairly just looks like a standard green spectrum. And then you have HICO. It obviously had a little problem in the blue parts of the spectrum being a little low, but certainly was showing some consistent features that if we look at the absorption spectrum for cryptophytes, which is down in this third panel, you could see we have some absorption due to phycoerythrin and chlorophyll and C2. But this weird resulting reflectance peak could not be modeled with hydrolite if we assume an absorption and a scattering. Any way you try to model this spectrum you see with this weird peak, you just can't get that because the absorption bands are not this small, and that amount of reflectance difference was too great. And so we started thinking, wow, this is an unusual peak, and it was pretty coherent across that whole region. And the one thing you know about mesodynium is it also fluoresces very bright yellow in the lab with the phycoerythrin and the chloroplasts. And so certainly when you have a concentrated, we see red, but they're also fluorescing this bright yellow peak in the waters at this time. So that really was our first, like, aha, if you put up a satellite, you might see something that you can't model with hydrolite or easily understand in the water. And so we came up with just a simple algorithm here. It was a continuum dip feature from 560 to 588 to look at this enhancement in the 580 part of the spectrum there. So it's looking here, this dip here. And so we then modeled this with the HICO satellite, and you see these patchy, very dense regions where you have this yellow fluorescence. Very much more detailed image than what you could see from Terra Modis fluorescence. And we could then link it to almost nearly 1 million cells of these cryptophytes that were producing in this part of the Long Island Sound. So this was kind of exciting, and this is why I'm so excited to put up hyperspectral sensors, because I think there's so many discoveries yet. So we wrote this paper, it was in PNAS, called "The Space Station Image Captures a Red Tide Ciliate Bloom at High Spectral and Spatial Resolution." And we were really psyched about this work. And then the funnier part is the Science News picked it up, and they put the title, "Scientists Identify Plankton from Space." And so we got a lot of weird press with people saying, what kind of plankton from space did you get? And where did the plankton come from? Mars or like outer galaxies? So, you know, just beware sometimes when you publish something, it may not be exactly picked up with the right words. I mean, I guess it got good press then, but it was a little different message. And there was, like, the Trekkie people were very interested in this paper. All right, so as I said, we're starting to fill it in. Now we're doing 570, 580 bands there. So now just my own personal research in Long Island Sound, I'm starting to fill in this table. And these are important wave bands that I think are important. And so you can see as you start working through this, like, we need to put up full hyperspectral sensors to discover just the true diversity of the ocean. So, you know, where would we like to go to next with this Long Island Sound? The graph there on the right shows the spectral, shows the different phytoplankton communities. So we have diatoms, dinoflagellates, cyanos, and cryptophytes, which, you know, we already see in those mesodiniums. So we'd like to sort of go finish the time series, continue just the base chlorophyll, but then really start to tackle phytoplankton composition and this diversity, and try to understand their contribution to carbon fixation and photosynthesis. Because as I showed on the right, I mean, we haven't done these kinds of studies, but clearly there's something going on. The cryptophytes, I don't know if our cryptophytes are similar to the ones in the Antarctic, but they don't do as much carbon drawdown as when there's these brown juicy diatoms in this area. So it would be nice to know then to take this, use our hyperspectral data and sort of tackle and tease out the phytoplankton composition, which is some work we're doing in the Antarctic now. Which can extrapolate to Long Island Sound as well, and understand better the carbon dynamics of this region. And then just the whole host, we're hoping, if you put up the sensors, we'll be able to kind of tackle a lot more of the applications from global change to ecological indicators like hypoxia and eutrophication, whether we're doing a better job in Long Island Sound, which regions need to be tackled more. We can't do that until we get better models. And I think I've shown you that really the bands matter on these sensors. And we for so long have been just putting up these standard bands to get chlorophyll, and they're not sufficient to really capture these dynamic waters we find in your region, which I know is very dynamic, and in the region in my backyard, Long Island Sound. So with that, this is from my sabbatical. I was very lucky to live in Bruges for a year, tackling North Sea science. And so with that, I want to thank you, and I'll take any questions.
Thank you, Heidi.
Thanks.
Presentation. So I'll open up the questions either in chat, or if anybody has a question, you know, you're welcome to, you know, unmute yourself and ask your question to Heidi directly.
Oh, Heidi, it's so nice. It's a scintillating presentation. I really enjoy it. So on that, I have two questions. For that Long Island Sound, you found in the winter that that there's a very little fluorescence with phytoplankton due to photosynthesis. I mean, low temperature. Is it just a local phenomenon, or it's a general?
You know, that's a really good question. I don't know if it's just the phytoplankton don't give off a lot of their light because they're such low light in winter. You know, they're just kind of keeping it. So there's not an excess to fluoresce like compared to summer. So then if that's the case, then that could be happening in many regions, right?
Yeah.
Where there's certainly the quantum yield of fluorescence, which isn't my specialty, but I think it does change with, you know, the dynamics going on with the light field in general. And it's something we should really also tackle. We're going to need to tackle for sure for new applications using fluorescence in the future. Now, I don't know if MODIS just couldn't have the sensitivity that there was fluorescing a little bit, and it's just all in that because it's such a low light and we don't have good sensitivity. It certainly wasn't fluorescing as much as in the summer, but it probably was doing a little bit, which we didn't see at all. And that could be because the gains are set so low, and it could all be in that first bin of kind of fluorescence that we can't differentiate.
My second question is on the mixotrophic the ciliate or at the bugs. The mixotrophic, they are not born at the mixotrophic. They have to acquire, right? Later, or they are born at the mixotrophic.
Yeah. Well, is it an obligate mixotroph, meaning do they have to do photosynthesis?
Mm-hmm.
I think they, I don't know. I'm not sure. Is that your question? Like, do they do this just when they can, or do they do it all the time?
Yeah, I just say it seems to me it's a really a symbiotic relationship. I don't know if that like mixotrophic. There this thing, if they reproduce, do they produce mixotrophic or just
No,
all?
they
don't, right?
Yeah, they don't. They enslave them, meaning what they ingest, they put them on their backs. Yeah, there's other pretty examples of mixotrophy that do quite a bit of photosynthesis as well. And I'm not the first one. Actually, Shubha Sathendranath has published back in the 80s or 90s on the color of some mesodynium blooms that she encountered as well with airborne imagery. So it's clearly not, a one-off thing that just happens. I mean, it is sporadic. That's why we need sensors to see it when it happens. But I think it's more common and probably contributes more to photosynthesis than we think.
Yeah.
I'd like to monitor it more. And now that we have some sensors up, that'll be interesting to see if we can look at this time of year and see how often we see. I was totally surprised by the yellow fluorescence. I was, like, it makes sense, but you just don't see it with your eye, right? So you just wouldn't have known it was there.
But do you sample sound regularly or just occasionally you do?
Yeah, right now we're not doing a whole lot there, so we'd like to do more. There is a AeroNet OC station in Long Island Sound by our colleagues down at CCNY, Alex Gillerson. And so and there's also sampling of the pigments that keep every month. But we don't have hyperspectral with those pigments, so we'd like to augment their monthly sampling to get the hyperspectral, so we can really tease out the phytoplankton dynamics. So that's future work. It seems a shame they go out every month and we don't do any sampling with them.
I see the hand of Thomas up. Thomas, do you have a question?
Yeah, thanks. Thanks, Heidi. First of all, do you think that those space plankton will overtake us over? And
think they
only kidding. What about tackling the hypoxia? You showed earlier in the presentation that on that west end,
yeah,
where they had the D grade for hypoxia.
Yeah.
Later in the HYCO picture, you did not see that mixotroph presence. It was more to the east. Is there any correlation between those two? And have you looked at that? It seems like maybe the diatoms are more prevalent in the western portion.
Yeah, so that's a really good. Tackling the hypoxia is a pretty complex thing because it's a pretty shallow estuary, so it has a lot to do with the direction the winds and the waves come, and if they happen to be together, you can get mixing, and if they're not, then you don't mix the oxygen down, so it kind of caps over. So there is this whole question of also allochthonous versus autochthonous, like how much is from, because you saw that the algorithms show that there's just enormous amounts of CDOM and other dissolved organic matter. So I don't know if it's just that the bloom happens, or is it because the water's warm, they stratify and we don't get mixing and all that organic matter is decaying down there. So it's still an open question, and we need sort of more monitoring down there to sort of close the budgets. I don't think it's sort of an active, ongoing several researchers, but I think we need to sort of harness all of our assets to sort of look at that question better. But I agree, it could be linked to the phytoplankton types as well. And I unfortunately haven't put it all in a model yet, but they're working on it. There's several, Jim O'Donnell and Craig Tobias, and there's others in the department that are really engaged in the hypoxia modeling. So, and the thing is, we don't know if it's better because climate change and shifts in, you know, patterns, or is it getting better because of management? And that's kind of a key issue. Are you studying oxygen in like low oxygen waters?
I am not, but I do have, we do have some observations for localized oxygen
Yeah,
in this area.
the sound is pretty shallow, so it's like 20 meters or so. So, you know, at some spots down to 40. So mixing is a big aspect to it. I see there's a question, Martin, and one other.
Voila. Yeah, so it's interesting that you showed that the hypoxia is actually the hypoxia events are being reduced near sort of close to Manhattan.
Yeah.
And there was actually recently an article in the New York Times that said that that the quality of living for all kinds of wildlife is improving in New York City. So then I thought, oh, that's interesting. Maybe they're related. So I only wanted to make that comment. It was just an article that appeared.
Yeah, they said the wildlife
yesterday or today or something.
Waters or the land.
Yeah, I didn't scan the article carefully.
There's like coy wolves. There's these scary coyote wolf things here that are really large, and they live in New York. Like they're doing well, and there's some here as well. And when you see them, they're like big coyote wolf like things, and they're super smart and shy. So I've read that they're doing really well in New York.
Okay.
The coy wolf, it is true, they did change some of the sewage treatment plants, which could have influenced the amount coming through that East River into Long Island Sound from New York. I think that did play a part in reducing water quality in that part. But I'm not positive it's been unequivocally shown. And also, the interesting thing about Long Island Sound I didn't say is it's right on that subtropical subpolar line where as far south as lobsters grow. But our lobsters have been suffering from a whole bunch of shellfish, shell diseases and other things. And so people for some time were saying, what's, changing? And I think now it's just really the waters are warming and it's no longer desirable habitat for the lobsters, and they're more being targeted by all sorts of pathogens. And not that there's suddenly a new pathogen, they've always been there, but it's just they're running at their limit with the warming. So lobsters are definitely moving out of Long Island Sound, and we're getting more subtropical. So we're at this transition zone, which is kind of interesting species-wise, and it's probably going to affect even the base of the food web. These kinds of different blooms that'll happen, and so yeah, it's a very dynamic area. And I can say I used to take my students to the nuclear power plant. You can say, why would you take like marine biology students to a nuclear power plant? But there is one, it's on Niantic, and they have this warm pool of water that comes out. So we would compare the intertidal of the organisms of the warm pool versus the cold pool. But turns out now Long Island Sound's become so warm in the summer that they can't use it to cool the power plant anymore. So they have to shut down just because the water's unequivocally warming. So clearly there's a lot of changes going on in this ecosystem.
All right. So, in the interest of time, we just hit noon.
It's one for me.
Well, here for us, yeah. So, if there are no more questions, I would like to thank you, Heidi, one more time.
Well, it was so fun chatting with you. Please shoot me an email if you have any questions, and I'd love to hear from you.
Exactly, that's what I was going to suggest. In a normal setting, Heidi would be here. We would be meeting, with you in the afternoon and so on and so forth. So in this setting, I encourage everyone, who has follow-up questions, to reach out to Heidi.
Is that little pub on the base still open? There was that little pub.
I don't know. I don't know if that's open or not. I'm not sure.
Cypress House? Yeah, I'm not sure either. Yeah.
Cypress House.
Yes. So
next time.
Next time.
Okay. Take care.
Okay, Heidi. Yeah.
All right. Thank you very much, and thank you everyone for joining us today.
Okay. Take care.
Have a good weekend.
Bye.